Welcome back to the deep dive. We're here to really cut through the noise of some complex research and give you the essential insights.
That's the goal.
And today, our focus is, well, it's on a really tough clinical challenge. Brain metastases, or BRM.
Yeah, it's devastating when solid tumors like lung, breast, melanoma, colon cancer, when they spread to the brain, survival rates really drop.
And the treatment options just become so limited, right? It's notoriously difficult.
Hugely difficult. We're talking about, maybe 30% of patients with advanced cancers facing this. And for, well, decades, treatment's been tough because the brain is just such a unique environment, kind of hostile, really.
And we've lacked that fundamental understanding of what makes these specific tumor cells tick once they get there. Yeah. The ecosystem they build.
Exactly. What makes them so aggressive in that specific place?
Which is why we dove into this really groundbreaking source material today. It's a comprehensive pan-cancer human brain metastases atlas.
Yeah, and this isn't the old way of doing things, grinding up the whole tumor, getting this sort of average smoothie view of the cells.
Right, the bulk analysis. This is different.
Totally different. This uses integrative single-cell RNA sequencing, cRNA-seq. Think of it like looking at every single ingredient in that smoothie, one by one, cell by cell.
Wow.
They pulled together data from 108 BRM samples, and crucially, they compared them to 111 primary tumors, the original tumors, or PTs, from the same lineage.
Okay, and that single-cell view, that's the game changer here.
Absolutely. It lets you see the really subtle changes, different cell states, shifts in composition, things you totally miss with bulk sequencing.
And we're not just looking for the average bad guy cell. We're hunting for those, rare, super specialized villains that actually drive the spread to the brain.
Precisely. The cells that somehow learn to sneak past the blood-brain barrier, which is no easy feat, and then actually thrive in this completely foreign territory. And this analysis, well, it really delivered. It didn't just find common survival tactics, but also identified, entirely new cell populations that seem to help the tumor grow and resist treatment.
Okay, let's unpack this then. We need to understand the fundamental changes, right? When a cancer cell makes that leap successfully to the brain.
Yeah.
So we'll look at the sort of shared blueprint for the malignant cells themselves. Then, how the brain environment gets remodeled around them. And finally, this new classification system that came out of the research.
Sounds good.
Right. Let's start with the main actors, the metastatic tumor cells, the MTCs. What's the shared fundamental hardware change? What gives these cells the edge they need to survive up there?
So the single most, I'd say, striking finding across the board was a massive conserved increase in something called chromosomal instability, or CIN.
CIN. Okay, what does that mean in simple terms?
Basically, the genome, the DNA blueprint inside the MTCs is far more structurally chaotic than in the primary tumor cells. It's like the cell's operating system is just constantly spitting out corrupted files.
Huh. More errors.
Yeah, lots more alterations, breaks, rearrangements, and paradoxically, this chaos seems to grant it rapid adaptability. It can change faster.
So it's not just a few key mutations. It's like the whole organizational structure is breaking down, but in a way that helps it.
That's a good way to put it. The fundamental organization is unstable, and the magnitude was significant.
How big a change are we talking?
Well, they measured the fraction of genome altered, the FGA, and in BRMs, it was more than double what they found in the primary tumors.
Double the genomic chaos.
Yeah.
Yeah.
And this wasn't just in one cancer type. It was a shared feature across lung, breast, melanoma, colon BRMs. It really confirms that high CIN is this essential shared adaptation tool, and unfortunately, it directly contributes to poor clinical outcomes, no matter where the cancer started.
Okay, so CIN is the chaos, the engine for adaptation. But what about the strategy? If they're surviving in the brain, they must need to sort of fit in, adopt a brain-friendly identity somehow.
Precisely. And that led them to discover this common survival blueprint. They termed it the neural-like metaprogram, or MP9.
A metaprogram, like a software package?
Kind of, yeah. It's essentially a shared set of functional instructions, a specific pattern of gene expression that helps the cells survive in a particular niche. And since they landed in the brain, the dominant shared program they switch on is indeed neural-like.
Wait a minute. If these are, say, lung cancer cells originally, does this mean they suddenly start acting like brain cells? Speaking the brain's molecular language.
Well, that's the really intriguing part. MP9 is strongly enriched in the brain metastases, no question. But they also detected it, albeit at a lower frequency, already existing in some cells within the primary tumors.
Ah, okay. So it's not necessarily learned in the brain.
It strongly suggests that metastasis isn't just random chance. It supports this idea that there's a small pre-existing group of cells in the primary tumor that already has some of these necessary neural-like characteristics, the sort of molecular toolkit, maybe the navigation skills to specifically find their way to the brain and set up shop.
That really reframes the whole process then. The brain isn't just a passive landing site. It's selecting for these specialized travelers that already have the right passport.
Exactly. The brain niche selects for cells predisposed to survive there.
So which genes are central to this neural-like survival program, this MP9?
Well, a couple of examples they highlighted are MACF1, that's linked to both CNS disorders and tumor progression, and another one called MALAT1. MALAT1 seems to help these metastatic cells sort of reactivate and also hide from immune T cells.
Okay, hiding from the cops.
Sort of, yeah. And beyond just MP9, these MTCs consistently flip the switches on the classic cancer hallmarks. They massively upregulate genes for proliferation, just growing faster, and angiogenesis building their own blood supply. And at the same time, they downregulate things related to immune inflammation, basically hiding from the immune system, and apoptosis, which is programmed cell death.
So turn on the gas, turn off the brakes, and put on an invisibility cloak.
That's a pretty good analogy, yeah. They turn on growth, turn off self-destruct.
Were there any particular, maybe surprising universal growth drivers that popped up across different cancer types?
They did identify 3 genes that were consistently upregulated, shared across the different BRMs. VGFA was one that's kind of expected for angiogenesis, building blood vessels.
Yep, standard villain.
PNN was another, involved in cell adhesion. But the unexpected one was PLCG2.
PLCG2, what's that usually known for?
Well, it's been studied more in the context of neurodegenerative diseases like Alzheimer's, but this study clearly links PLCG2 activity to BRM across multiple cancer types.
Interesting.
Which suggests it could be a really critical, maybe even universal therapeutic target, something you could potentially hit regardless of where the cancer started.
Okay, let's do a quick recap here. We've learned that the cancer cells that make it to the brain, the MTCs, share some core features.
Right.
First, their genomes are way more chaotic, double the instability, this high CIN.
Yeah.
Second, they activate a common survival plan, a neural-like program called MP9, which might even pre-exist in the primary tumor.
Correct.
And third, they crank up growth and blood vessel formation, partly using this surprising gene, PLCG2, while shutting down their own death signals and immune visibility.
That sums it up nicely. Those are the core features of the malignant cell itself.
But you know, a malignant cell is only as strong as the environment it manages to create or co-opt. So let's shift focus now to that terrain, the tumor microenvironment, or TME, in the brain. How much does the brain fundamentally change the game, the ecosystem around the tumor?
Oh, it changes it drastically. But interestingly, it does so differently depending on the original cancer type.
Ah.
So the researchers came up with a metric for this called the remodeling index, or RI.
Okay, RI. What's it measure?
It basically quantifies how similar the mix of cells, the cellular composition of the BRM is, compared to its matched primary tumor, the PT.
So high RI means big changes happen during metastasis.
Exactly. A high RI means the metastasis forced a huge remodeling of the local brain ecosystem. A low RI means the TME state in the brain was largely just inherited from wherever it came from.
Okay, can you give us an example of extreme remodeling? Who completely changes their identity when they arrive in the brain?
Breast cancer, particularly the aggressive triple-negative type, TNBC, that showed the highest remodeling by far, RI up to like 0.721.
Wow, that's high. What changes?
Well, the primary TNBC tumor often has, a decent number of immune cells kicking around, but once it metastasizes to the brain, it shifts completely into what they call an immune desert.
So the immune cells disappear.
Yeah, the brain environment essentially seems to strip away that immune infiltration that might have been present originally. It becomes very cold, immunologically.
So for TNBC, the brain actively helps create an immunosuppressive shield. What about the other end, the ones that inherit their environment?
That would be colorectal cancer, CRC, and small cell lung cancer, SCLC. They showed very low remodeling indices.
So they arrive and basically keep the same neighborhood feel.
Pretty much, yeah.
Right.
For these types, the TME composition was largely inherited. SCLC, for instance, is often an immune desert already in the lung.
Okay.
And it just stays that way when it gets to the brain. So for these cancers, the TME wasn't necessarily induced by the brain. It was maybe a characteristic they already had that allowed them to succeed in getting there in the first place.
Interesting distinction. But regardless of how it gets there, inherited or remodeled, the resulting TME in the brain is generally immunosuppressive.
Yeah, overwhelmingly so, yes.
So where does that suppression actually come from? Are the T cells, the usual fighters, just failing? Or are other cells actively suppressing them?
It seems to be both, but the TME gets dominated by immunosuppressive cell types, particularly myeloid cells and stromal cells. When they looked closely at the lymphoid cells, so the T cells and NK cells, the natural killer cells, they found notably lower effector states.
Meaning they weren't really activated.
Right. Fewer T cells seem to be actively fighting or even in an exhausted state, which you sometimes see after a long battle. Overall, T-cell activation just seemed weaker in the BRM compared to the primary tumors.
So the immune foot soldiers are there, maybe, but they're largely sitting on the sidelines, ineffective or suppressed.
Exactly. And worse, the macrophages seem to be actively helping the tumor.
Ah, the macrophages.
Mm-hmm.
They can be good guys or bad guys, right?
Yeah.
M1 versus M2.
Precisely. And in BRM, they found significantly fewer M1 macrophages. Those are the pro-inflammatory tumor-fighting ones. But they found much higher levels of M2 macrophages.
So the bad guys in this context.
Yeah, the M2s are generally anti-inflammatory, tumor-promoting. They help with remodeling, angiogenesis, suppression. So this M1 to M2 shift essentially flips the macrophage population from potential defenders into tumor collaborators.
Any specific type of M2 that stood out?
Yes, a subset they called MO4. These MO4 macrophages were associated with hypoxia, low oxygen, and also with vascular functions, building blood vessels. And this specific subset was highly enriched in the BRM-TME key, really pushing that immunosuppressive pro-tumor environment.
Okay, let's zero in on that vascular system then. The blood supply. Did the single-cell analysis find anything unusual about the new blood vessels the tumor gets the brain to build for it?
This was actually one of the most surprising findings, I think. At the interface between the blood vessels and the tumor, the BTI, the analysis identified a really distinct subset of endothelial cells, the cells that line the vessels. They called it EO3.
EO3. Okay, what's special about it?
Well, first, it was way more abundant in the BRM compared to the primary tumors, like a threefold increase. But critically, these EO3 cells were somehow remodeled to show features of lymphatic endothelial cells, LEC-like features.
Wait. Lymphatic vessels, like the ones that drain fluid and traffic immune cells, aren't those like famously absent from the normal healthy brain parenchyma?
Exactly. The central nervous system is generally considered to lack a conventional lymphatic drainage system. So finding these tumor-associated vessels taking on lymphatic characteristics, it's highly unexpected. The tumor seems to be building a sort of counterfeit lymphatic system inside the brain.
Wow. What kind of lymphatic markers did they express?
Things like FLT4, also known as VEAFR3, and LYVE1. These are typically found on lymphatic vessels elsewhere in the body, not normal brain blood vessels.
And what's the implication of having these weird LEC-like vessels?
Well, it's not good. The presence of this EO3 subset is highly correlated with poor patient prognosis. And maybe even more crucially, this specific vascular subtype is linked to resistance to both immunotherapy and chemotherapy drugs.
So it's not just a weird structural thing. It's actively involved in treatment failure.
It appears to be an active player in therapy resistance, yes. A really significant discovery.
Okay, second recap time. We've moved from the cancer cell itself to its neighborhood. The TME in the brain is generally immunosuppressive. This happens either because the cancer brought that property with it, like SCLC, or because the brain environment forces a change, like in TNBC turning into an immune desert. This involves fewer active T cells.
And a big shift towards tumor-helping M2 macrophages.
And crucially, the tumor builds this weird, unique blood vessel system. These EO3 cells that look like lymphatic vessels, which are normally not in the brain. And these vessels are linked to poor prognosis and drug resistance.
You got it. That's the remodeled resistant environment in a nutshell.
So we have all this incredibly detailed molecular and cellular data, the CIN, the MP9 in the tumor cells, the M2 shift, the EO3 vessels in the environment. How do we bundle this up into something clinically useful, like a classification system?
That's exactly what the researchers did next. They used all that cellular abundance data, basically, the percentages of tumor cells versus immune cells versus stromal cells in each sample, and performed clustering analysis.
To see if natural groups emerged.
Precisely. And they identified 5 distinct ecological types, or ecotypes, of brain metastases. This gives us a new way to classify them, moving beyond just the primary cancer origin and looking at the actual ecology of the metastasis in the brain.
Okay, what are the 5 ecotypes?
Right, there's one. Desert type, D type. This one is overwhelmingly dominated by the tumor cells themselves. Very few immune or stromal cells. Think SCLC, TNBC often fall here.
The immune desert we talked about.
Exactly, inherently cold. 2. Lymphoid-enriched type, L-type. As the name suggests, high infiltration of lymphoid cells, T cells, and K cells. 3. Myeloid-enriched type, M-type. This one is dominated by myeloid cells, those macrophages we discussed, maybe other related cells. 4. Stromal-enriched type, S-type. A higher proportion of stromal cells, the structural or connective tissue type cells. 5. And finally, balanced type, B-type. Kind of a mix, no single component totally dominates.
This sounds incredibly practical, like for precision medicine. If you could figure out a patient's BRM is a D-type, you probably wouldn't give them a standard immunotherapy, right? Because there are no immune cells there for it to work on.
Exactly. You'd potentially save them the toxicity of an ineffective treatment. It really helps guide intervention strategies.
Any examples of how this links to known features?
Yeah, they showed a nice example with lung cancer BRM. Patients who had specific EGFR mutations in their tumors were much more likely to have the M-type ecology, the myeloid-enriched type, about 45% of them. Compared to patients with wild-type EGFR, where only about 30% had M-type.
Okay, so EGFR mutation status influences the TME ecology in the brain. What does that suggest therapeutically?
It suggests that maybe those EGFR mutant patients, because their BRM-TME is so full of myeloid cells, might benefit more from combination therapies that specifically target those myeloid cells, perhaps alongside their EGFR inhibitor, rather than just relying on, say, T-cell checkpoint inhibitors alone.
Tailoring treatment based on the actual cellular neighborhood within the metastasis. Makes sense. Okay, final piece. For patients whose tumors maybe don't fit neatly or maybe need a broader approach, did the analysis point to any single master key target, something that cuts across these different ecotypes?
They looked really closely at the communication network between the cells. You know how cells talk using ligands and receptors?
Signaling pathways.
Right. And they identified one specific interaction pair that seemed to be broadly shared across many different BRM types and ecotypes. It's the Nectin-2 TIGIT immune checkpoint axis.
Nectin-2 TIGIT. Okay, TIGIT is definitely a hot target in cancer immunotherapy research right now.
It absolutely is. And this interaction, Nectin-2 on stromal cells like fibroblasts or those EO3 endothelial cells binding to TIGIT on T cells, seemed to happen consistently in the BRM samples.
So finding this is a common vulnerability, specifically in BRM, that suggests targeting this axis, maybe with TIGIT inhibitors, could be a widely applicable strategy.
That's the implication, yes. It provides a strong data-driven reason to explore this Nectin-2 TIGIT blockade, likely as part of a combination therapy, as a potentially universal approach for treating BRMs, complementing other treatments, and hitting a weakness shared by many of them, regardless of their origin or specific ecotype.
Wow. Okay, this deep dive has really given us an essential roadmap, hasn't it? Moving from these incredibly detailed single-cell insights towards potential clinical actions.
I think so.
We've defined the shared molecular machinery inside the cancer cells, the CIN, MP9, PLCG2. We've categorized how the brain environment changes using that RI metric. And we've got this new classification system, the ecotypes, pointing towards more tailored therapies, plus this potential universal target in Nectin-2 TIGIT.
It's a huge leap in understanding. But there's always a but, isn't there?
Always. What's the limitation here?
Well, the researchers themselves noted this. This entire amazing atlas is based on identifying what cells are present and what genes they're expressing. It's like, counting all the ingredients in the recipe.
Okay.
But architecture matters. Location, location, location.
You mean they know who the cellular players are, but not necessarily where they are sitting in relation to each other within the actual physical structure of the tumor?
Precisely that. We know, for example, that the EO3 LEC-like cell is immunosuppressive, and we know T cells are suppressed. But the next big challenge is connecting that knowledge to the spatial arrangement.
How does the physical location of that EO3 cell, right next to a T cell versus further away, actually change the outcome? Does that proximity determine resistance?
That's the question. How does the 3D architecture of the BRM niche influence these interactions and ultimately affect how well therapies work? It's about understanding the context, not just the components.
Right. It's the final frontier, maybe spatial biology, bringing that location data into the picture.
It's where the field is heading, for sure. Integrating single-cell data with a spatial context.
So a final thought for you listening. Considering everything we've discussed, the chaotic cells, the tricky environment, the weird blood vessels, what cellular interaction in its specific physical spot within the tumor do you think holds the key to finally unlocking better treatments for brain metastasis? Something to mull over.
Definitely food for thought.
Join us next time for another deep dive.